Good morning, everybody. So I am Joan. I am a co-founder and CEO at Flomics Biotech. Today we are going to present a webinar about genomic services and collaborations at Flomics. We are going to wait just a few more minutes for the rest of the attendees to join, and we will start very soon. Well, so 5 minutes passed. I think we can start slowly. I ask all the presenters to mute themselves when they are not speaking for a better audio quality. And for all the attendees, feel free to write your questions in the question and answers area, and we will try to go over them at the end of the seminar. In case we don't have time to go over all the questions, we will get in touch with you directly, and we will make sure that everything is clear. So yeah, we are about to start. Luke, maybe you can change the slide. Uh, so as I said, I am João, one of the co-founders of Flomics. Today with us we have Luke Cabuz, the head of contract research services at Flomics, and Mercedes Serrano, pediatric neurologist and principal researcher at the Hospital Sant Joan de Déu in Barcelona. Flomics, we are a biotech company also based in Barcelona. We thank BioCat and all the organizers of the Barcelona Health Innovation Week for the opportunity to reach to the audience and present a bit about our technologies and services. And with this, I will act as a moderator. So as I said, if you have questions, please write them in the question area, and I will try to go over them at the end of the seminar. So I pass the word now to Luke. The floor is yours.
Okay, thank you, Joan. Okay, so without further ado, I will start. So I will explain a bit the genomic services of Flomics, and then I, together with Merche, will explain a case for biomarker discovery of cerebellar atrophy. Okay, so first of all, Flomics was founded in 2018 in the PRPB, and recently we have moved to Cosimbio Labs in Poblenou in Barcelona, which is this image that you can see in the bottom, where we have, this is a co-working space with a wet laboratory. At Flomics, we are specialists in the application of RNA to different fields, such as oncology or metagenomics, and we provide tailored omics and bioinformatics solution. And actually, we recently acquired our first NGS sequencer. So Flomics is divided into 4 different parts. First of all, the genomic services, where we offer our expertise in NGS and molecular biology. We also have the custom bioinformatics services, where we adapt the bioinformatic analysis to the needs of the project. We also have Stratus, which is a cloud software for independent bioinformatic analysis. And we have the Flomics liquid biopsy solution, where we scan the cell-free RNA for the screening and monitoring of complex diseases, such as cancer. Our genomic services, so at Flomics, we offer the genomic services as a complete solution. So we can receive directly the sample, and we can do the genomics and bioinformatic analysis, doing several quality controls at both steps, tailored to each of the projects. So we are very flexible in that sense, and we can adapt to different sample requirements, since we are used to work with plasma, which is a very difficult sample. And then we have a wide range of well-optimized experimental protocols. And we can also customize the data analysis and the reporting of the results. So due to this flexibility, we have partners with different institutions in a broad range of projects, such as liquid biopsies for complex diseases, as we will explain with Merche later, to the detection and surveillance of SARS-CoV-2 variants in wastewater samples, and well, everything in between and beyond that. So a workflow for NGS can begin, for example, with us receiving the samples and performing all of the wet lab procedures with quality control, sample preparation, doing the nucleic acid extraction, and doing the libraries, and then transforming these nucleic acids into data via sequencing. Then, when we have this data, we can also do the bioinformatic analysis to understand it, doing the primary, secondary, and even tertiary analysis, and ending with a final report of the results that we found. We can also you can also work with Stratus, which is bioinformatics as a service, where you can upload directly sequencing data and run some of our custom pipelines with some modifications. And this can grow without limits. So we can process any amount of samples in less than a day with some pipelines that we have customized in-house. Stratus is a platform that is in the cloud and is web-based. So there's no need for bioinformatics training. You directly can run some custom pipelines that we have done. We have optimized, and you can select them. You can upload the data, and then you can run a workflow with a very reproducible way. So you can obtain some results quick, and it is very flexible. However, I won't go into much detail because my colleagues here, Julian and Christina, did a webinar of Stratus on Tuesday, which is recorded on our website. So if you have the time and you didn't go to the webinar, I highly recommend to go there. So we have two types of collaboration models. We can work in a standard collaboration where you come to us with a project and then we together apply to funding. Then each party contributes with their own resources to the project. And then in the end, we share the intellectual property and the publication that can arise from it. We can also have another collaboration, which is as a service provider, where it's the same. You came to us with a project, and then we do some part of the project as an external party, and then the research center owns their own IP. So I will go to some of our expertise and some relevant projects that we have worked on. So first of all, our expertise lies in cell-free RNA. Cell-free RNA is the RNA that is circulating on the plasma, and it can act as a biomarker for several diseases, several complex diseases. There have been several papers talking about this, mainly in the areas of cancer, but also in other different complex diseases. And this is a field that has been rising during the last few years. And if you're interested, we have a review of the challenges in this field, in the field of cell-free RNA for biomarker discovery, where we go into the challenges of this field and how to optimize a protocol, which are the most important steps to optimize. And this is something that we also have developed in-house. And now we have an optimized and production level cell-free RNA protocol, starting from the plasma isolation and all the way to the gene quantification, with extensive quality control at each step to ensure that the results are reproducible and have the best quality possible. So using this protocol, we did some pilot study in colorectal cancer when we obtained a blood sample from patients and controls, and we quantified over 20,000 genes, and running through different machine learning methods, we obtained a disease signature that has a score for each of the samples. With this signature, we obtained very impressive results with 86% accuracy in the validation cohort, and with 100% sensitivity. This means detecting correctly all of the colorectal cancer patients, even in the early stages. Right now, we have an ongoing study with 5 different types of cancer and more than 1,000 patients to validate these results and to perform the same type of study with other types of cancer. So, as other examples of projects that we have done is this one with DNA variant identification and interpretation, where we obtained some oncology samples, so samples from tumors, and we did the classification of the variants. This is something that is really important in the clinic. And also we did the clinical interpretation. So knowing if these samples, if these mutations, were benign, possibly damaging, or probably damaging. Also, going even further with our report, we say which of these alterations are the most relevant using a database, 2 databases, one called Cosmic and the other one called Civic. And we go even a step further and we provide evidence of the type of therapy that can work best for these type of mutations, so that the clinicians can go directly and select the type of therapy that will be the best. Also, another need that was in the field, so we had a client that wanted to do metagenomics analysis, but they wanted to do it in a reproducible manner and in an automated way, let's say. So they didn't want us to do a specific project, but they want to directly run a pipeline, but they didn't have a bioinformatician in the team. So what we did was we created a pipeline that did the metagenomics analysis, the phylogenetic classification, etc. And then we uploaded this pipeline into Stratus so they can work directly uploading the samples into Stratus and obtaining the results. So the interactive reports and also the file with the different results of the pipeline. Again, this is something that my colleagues presented on Tuesday, so I won't go into much detail. So I will go further. So at Cosimbio, we have a fully equipped laboratory with brand new machines. We have the Illumina NextSeq 2000 and Maxwell robot for automated RNA extraction. And right now we are processing a lot of samples. So I wanted to highlight that we have a multidisciplinary team, mainly in the fields of bioinformatics and molecular biology, and well, with some of the lead experts in the field. So without further ado, I will give the floor to Merche. Merche, if you want to present.
Yeah, thank you, Joan. It's my great pleasure to join you today, and particularly to talk about the cerebellar condition that we've been studying since more than a decade, and this wonderful collaboration. Next, please. To put the audience in context, pediatric cerebellar atrophy is a more or less frequent condition that we see in the consultations of child neurology. In our hospital, in fact, which is a tertiary care hospital, we have more than 300 patients with this condition. And at present time, despite the application of next-generation sequencing in the clinical practice, up to 40%, near 40% of the patients still have no diagnosis, despite showing this clear abnormality in the magnetic resonance imaging and the disability. And among the disease causing cerebellar atrophy in children, we can recognize a paradigmatic disease that is a metabolic condition caused by the deficiency of phosphomonoamidase that you can see written here, and is a congenital disorder of protein glycosylation. And in fact, it's the most frequent CDT, congenital disorder of glycosylation. Next, please. PMN2-CDD is a rare disease with less than 1,000 patients diagnosed worldwide, and it's a genetic condition caused by mutations on gene PMN2. And patients normally develop a multi-organic disease, but cerebellar atrophy is a hallmark. And as you can see in the MRI on the left for this patient, there is a shrinking, there is a loss of volume on the cerebellum, particularly affected in the central nervous system. But unfortunately, when symptoms appear, the disease is irreversible. And next, despite the efforts of the international clinicians networks that we have not been able to find a genotype-phenotype correlation, which is a major limitation to give information to the families when the child is diagnosed. So we are not able to give more information than the confirmation of the disease. And on the other hand, we have no reliable or accessible laboratory biomarkers to monitor the evolution, to inform about the prognosis, or even more importantly, to evaluate our interventions, particularly pharmacological interventions. However, we feel that there is an urgent need to find objective biomarkers, since some promising therapies are developing and the clinical trials are on. And we have a particular difficulty in pediatric patients, and it's that only little sample volumes of blood can be taken out because of ethical reasons and because the whole amount of blood volume in their body is little. Duke?
Yes, so here is where we started collaborating. So what we did, in the Hospital Sant Joan de Déu, they obtained plasma from 29 patients and 68 controls. And then they sent a sample to us, and then we did the hemolysis quality control, which is something that we do always for plasma. And then we performed the microRNA isolation and library preparation with the respective quality controls. Then we sent the samples to sequencing and obtained the sequencing data. Then we also in-house processed this data using a state-of-the-art pipeline, and then we split the data into training and test sets, doing 80% of the data going into training and 20% into the test. And then we performed different kinds of filtering using for example, the differentially expressed genes, and 5 different types of machine learning algorithms to end up with a total of 15 models that we evaluated using this test set. Then we obtained a final model with high accuracy. So going straight to the results, we found that in the differential expression that there were very few differentially expressed genes. This is something that we know could happen because the biological variability within the group of patients was very high, because this was a very rare disease, and we had patients with the age ranging from a few months to adults. So even with this biological variability, we were able to find 6 microRNAs that were differentially expressed, with one being especially highly significant, as you can see here in the top left. That is very important for this disease, but we will talk later about this. So the result that we obtained is that this plasma microRNA signature had very high accuracy in all of the cases using this cross-validation. However, we found that the signatures had quite high variability. However, we found especially one signature, which is this one, using an algorithm called XGBoost, which I won't go too much into detail about this, but that had very, very high accuracy both in the training and in the test cohort. As you can see here in the ROC curve in the top right, this signature had an AUC higher than 0.9, both in the training and the test set, which are extremely good results. So, Merche, if you want to continue.
Yes, as you have said before, there is a microRNA which is much differentially expressed in patients and so high weight in the signature. And that central microRNA, next please, it is known to be implicated. Next slide, please. Thank you. Is known to be implicated in different cellular and biological pathways, and many of them are related to neurological condition, and particularly to our condition that we are studying. Next, please. In general terms, all those microRNA included in the final selection have relevant and already known roles in the different organs and systems that, in fact, are affected in PMM2 CDG. And going back to that top microRNA, it is known to be implicated not only in neurological disorders, but also liver tissue, which is also primarily affected in PMM2 CDG. So just to conclude, following the FDA definitions, a biomarker is a measurable indicator, objective indicator of some biological state or condition, but may have different roles and functions. And why these different functions are so relevant for us working in pediatric conditions? Because many patients with genetic neurodevelopmental conditions improve due to the natural process of neurodevelopment itself. And then it is difficult to monitor exactly the disease evolution separately of this natural process of neurodevelopment. Then we can apply our discovery in the diagnosis, but at present time, to be honest, it might be limited compared to the golden standard that is the next generation sequencing, the molecular confirmation. But it may be truly useful to identify a subset of patients that will show different phenotype or different prognosis to inform the families. Since, as said, there is no genotype-phenotype correlation today. Moreover, our findings aid us to unravel which biological pathways may be impaired and may help us to find new potential therapeutic targets. And importantly, our discovery might help to monitor natural history of disease or to evaluate therapy efficacy, since magnetic resonance is not much informative for evolution once we see the atrophy, stays in atrophy, and then the pediatric neurological patients show this natural improvement, and they normally are not able to fulfill the clinical scales that we use in adults, for instance. So they will be really useful in these purposes. We want to thank you so much for your attention, and Duke, if you were to conclude.
Yeah, okay. So right now we have finished the explanation. So we have our Q&A. Joe, if you want.
Yes. So now there's time for some Q&A. Please write your questions in the Q&A section, and we'll try to answer them, as I said. But before we start, I have a question to all of you. Let me start the poll here. As you know, we at Flomics have a wet laboratory, but also bioinformatics capacities. And we would like to know what are your main struggles, what is your main bottleneck in terms of research, or which are the field that you could get more help with bioinformatics or wet lab work. I know it's very, very general, but please answer this question. And while you answer, I will start with the first question that we have here today. So we have one question that I will read. Is there a minimum sample requirement for a type of analysis similar to the cerebellar atrophy project?
Okay, so it's a good question. So it really depends on the type of project that you want to run and the type of results that you expect. So for this type of projects, if we do some kind of machine learning, the number of samples will be higher. But for example, for a pilot study with doing some differential expression to see if there are some biomarkers that could be useful for this disease. For example, we could do 10 samples of patients and 10 samples of controls. We could obtain some first results that then we can do some further studies with a higher number of samples. But for a pilot study, for example, I would say that 10 samples of patients and 10 samples of controls could be okay.
Okay, thank you very much. So regarding the poll that I just shared, I can see that 60% of you have more struggles with the bioinformatics, 40% with the wet lab work. So, well, the thing that is important is that we can help you in both of these fields. So whatever you are struggling with getting the data analyzed or to get the sample processed, please get in touch with us, and we will try to help you the best we can. and also to understand a bit more the fields of research and which type of molecules or omics disciplines you are more interested at. We are now also asking if you are more interested in DNA, RNA-seq, metagenomics. Please answer with your favorite field of interest, and I will go for the second question we have today. How long does it take to process a plasma sample into sequencing data?
Okay, so well, it really depends on the number of samples that we have to run in parallel and the number of projects. But in general, we can obtain some results within 2 to 3 weeks if we run only a few samples. So for a project like the one from Sant Joan de Déu, with we got to obtain some sequencing results, not the results of the data analysis, within a few months.
Okay, and what about the sequencing? What about the data interpretation? What is the time frame? Can you also add up on that?
Yes, so it really depends, because sometimes there is more of an exploratory analysis. Then normally we do some first results, then we share with the research center that we're working with, and then we decide on how to move forward. It really depends. Doing some basic data analysis can be done between in 1 week, for example. But then if we do some machine learning, which is more difficult, let's say, it could be a few more weeks.
Okay, thank you. So we have one more question here. It starts with congratulations for the very interesting webinar. So congratulations, Luke and Merche. The question is, are you collaborating with different biobanks in Spain to obtain more samples to study cerebellar atrophy, or are you planning to do it in the future? Thank you.
Okay, so Merche, maybe you can answer this question.
Yeah, thank you, Laura. It's an interesting question. This data that we have shown is just a pilot study, and the next study that has been also financed by the Instituto de Salud Carlos III is the next step. So now we are working in a multicentric project, obtaining from the different patients living there this kind of samples. But yes, we are open to any collaboration, and everything is welcome.
Thank you, Merche. So I think we should wrap up. We're aiming to have a 30-minute session, and we are over the time. So thank you very much for everybody that participated. Thank you, Luke. Thank you, Merche, and thank you for all the attendees. As we have been saying, feel free to get in touch with us at Flomics. We are very interested into expanding our collaborations and to find a way to help you boost your research and to act as an extension of your own laboratory, of your own team. With this, I say goodbye, and I hope to see you soon. Bye-bye.
Thank you. Bye.